So in case of immune system cells, so there are some of the cells that are myeloid cells and the lymphoid cells. Lymphoid cells only the T cell, B cell, and the dendritic cell. Sorry, NK cell. These are the lymphoid cell, and the most of the cells are myeloid origin, like neutrophil, macrophage. They are the myeloid cell origin. So myeloid origin is actually these are the cells granulocyte. These are the myeloid origin. So you need to know if each and everyone's function. Each and everyone's function you have to know. I guess you all know already the what are the function of each cell. Like eosinophil, they have the role in the parasite, and which immunoglobulin actually take part in the parasitic killing. IG, IgE. IgE is the um, take part in the parasitic killing. Like this is the IgE, and whenever IgE binds, this is the FAV portion of the IgE. It will bind to the parasite, and the FC portion will bind to the FC receptor of the eosinophil, and ultimately eosinophil will release some of the some lytic enzyme and kill the parasite. So the parasitic infection, in case of parasitic infection, in case of allergy, the eosinophil takes part. In case of basophil and mast cell role is also in the allergy. And so these are the type 1 hypersensitivity, they usually takes place, basophil and mast cell. So cells, professional antigen presenting cell, we have already discussed macrophage, function of the macrophage, very important function of the macrophage. Apart from phagocytosis, macrophage actually causes the other functions too. If we see the picture of a macrophage, they have a lot of receptor, no? MAC1, MAC2, then they have other receptor like toll-like receptor. We have already seen what is the function of this toll-like receptor? To bind different type of pathogen associated molecular pattern like flagella, like LPS, and these are the function of the toll-like receptor. They have some scavenger receptor. They have some SC receptor of the IgG, so that whenever a bacteria or anything that bind by the antibody, the SC receptor of IgG will bind with that, and ultimately macrophage will really clear the thing from the body. So macrophage is actually the not only the phagocytosis take place, like but they also remove the um, um, other cells, other destroyed cells from the body too, and they also take part in the repair system of the body. So they have two type of activation. We have already said that M2 activation and M1 activation. M1 is the classical activation, and they will activate the T helper cell 1 inflammation and repair. There is a microbicidal activity and anti-tumor activity. And in case of M2 activation, there is a tissue repair and some other activities too, tumor promotion, T helper cell 2 activation. So the function of the macrophage is not only the phagocytosis or antigen presentation, there is some M2 activation or M2 function too. And T cell, B cell, so what will be this answer? Oh, it is written on already. Memory cell are present in case of, actually T cell and B cell will be the cell of adaptive immune system, and these cells have the memory. Right? So memory cell is a part of adaptive immune system. T cell, B cell is a part of adaptive immune system, and that presence that does the memory. Difference between the T cell and B cell important. We have already discussed what is the humoral immunity. What is the humoral immunity? What is the cell-mediated immunity? What are the cells? CD4 cells, CD8 cells, and what are the types of CD4 cell? What are the types of CD4 cell or T helper cell? T helper cell is T helper cell 1, then T helper cell 2, then T helper cell 17. Huh? T helper cell 1, what is the function of T helper cell 1? Cell-mediated immunity. And function of T helper cell 2 is the to produce humoral immunity or to generate the B cell. So these are the function you need to know. Answer this question. Main function of helper T cell. Is it true or false? Anyone? Can you
True.
hear me? Acha. Activation of the CD8 T cell via interleukin 2. Okay, let's see. Activation of B cell via interleukin 2. True or false?
False.
What will be the answer?
Interleukin 4.
4, 5. Right. Secret interleukin 2.
True.
True. Where this interleukin 2 goes into autocrine fashion, they will act that helper T cell also have. Activate natural killer cell. True or false? So let's check. Activate macrophage through gamma interferon. True or false?
True.
True. It's true. Natural killer cell works alone. They do not need the helper T cell. So this is the question or residency. We need to know a basic, clear concept because the knowledge will be required in the future. So cytotoxic, we have already discussed what are the role of cytotoxic T cell to kill the virus infected tumor cell macrophage and also the mismatch transplant cells. And then NK cell, role of NK cell is also the same. Role of NK cell is also the same. We will discuss what is the role of NK cell. Cytotoxic T cell are involved in the following function. Lysis the virus infected cell, true or false? True. Antibody production, true or false? Cytotoxic T cell, antibody production, true or false?
False.
Good. Graft,
True.
good. Tumor,
True.
good. Antigen presentation,
False.
good. So this is the picture. Already you have discussed the this is the picture, and now the question turns. This is the antigen. What is the chemical nature of this antigen?
Protein.
Hmm. T cell only recognize protein. This is the antigen presenting cell. What is the MHC? Which type of MHC is this? MHC 1 or 2?
2.
MHC 2. Antigen presenting cell can have MHC 1. Is it true or false? Antigen presenting cell have MHC 1. Is it true or false? Antigen presenting cell can have MHC 1. Is it true or false? Anyone else? True. Because antigen presenting cell is a nucleated cell and antigen presenting cell is a nucleated cell. So all nucleated cell present have MHC 1. MHC 1 is present in all nucleated cells. So antigen presenting cell have both MHC 1 and MHC 2. Both MHC 1 and MHC 2. Acha. So these are the cytokine. Cytokine. So helper T cell will help either differentiation of B cell. Helper T cell will help either activation of cytotoxic T cell or generate memory cell, or they will help the macrophage. So they will actually help the other cells to do work properly. Sometimes the B cell will be directly activated without the help of T cell. Without the help of T cell, antigen will bind to the B cell, and B cell will produce antibody. What is the chemical composition of this antigen? What is the chemical composition of this antigen when only B cell will be activated and produce antibody? Chemical composition of that antigen, protein or polysaccharide. Protein or polysaccharide. This is T independent antigen. This is T independent antigen. Or B cell activated sometimes without the help of T cell. That is T independent activation. So which one is better? T dependent activation of B cell or T independent activation of B cell? Which one is better?
T dependent, sir.
T dependent. T dependent activation is better because whenever B cell develops with the help of T cell, then the antibody is generated or T dependent generation antibody. What are the antibodies produced? All type of antibody. All type of antibodies are produced. We need all type of antibody for so many function. Another one, components of cell-mediated immunity. CD4, true or false?
True.
True. Memory, T cell.
True.
Let's check. Platelet, basophil, false. Na? They cannot take part. Macrophage, true or false?
True.
True, because they need to present. They need to present the cell, and that is true. Good. Another question. Cell-mediated immunity is the main host defense against Klebsiella. True or false? False. Klebsiella is extracellular. Pneumocystis jirovecii. What do you think? True or false? Okay, let's check. MTB,
True.
Candida. Let's check. Let's check.
False.
Uh, hmm. So bacteria, those are actually extracellular, but virus, fungus, MTB, facultative, they are the intercellular. So that will actually activate the cell-mediated immune system. We have already discussed the regulatory cell. Just last one, the NK cell. Actually, NK cell, the function of the NK cell is to kill the virus infected cell, tumor cell, and they will kill they will kill the cell with the help of cytotoxin and perforin. They will release some of the cell that will we are not going through this. So the natural killer cell is the cells of innate immune system, and they can take part as the ADCC, and their function is just like CD8 T cell. So the question is, suppose a virus infected cell um, will present the cellular antigen to the CD8 T cell with the help of MHC1, and then CD8 T cell will kill the virus infected cell. This is how cytotoxic T cell kills a virus infected cell with the help of MHC. Sometimes what virus do? Virus downregulate the expression. Virus downregulate the expression of MHC1. Then on that time, natural killer cell will kill the cell virus infect that do not have the MHC1, that will kill the natural killer cell. So natural killer cell actually can bind. It will make more complicated. So I'll discuss this another time. Do you have any problem about the ADCC? Do you have any problem with the ADCC? This can be come in the written. Huh? What is ADCC? That means this is also a bridge between the innate immune system and adaptive immune system. NK cell is a part of innate immune system. Whenever the antigen binds with the antibody, which is a part of adaptive immune system, then the antibody have the FC receptor, and the NK cell have FC receptor, and the antigen binds with this FC receptor, and now NK cell will release the perforin and granzyme and ultimately kill the virus infected cell. So this is called antibody dependent cellular cytotoxicity. And cells that take part in the innate immune system, CD4, apart from CD4, all of the innate immune system. Lymphoid organ, that's it from now. We have discussed the positive and negative selection, antigen, antibody. I have discussed before. Do you have any question now?
No, sir.
Okay. Anyone else? Do you have any question? Immune diagnostic tests also. Some of you have actually a question about the immune diagnostic test. What was the question?
Yes, sir, I asked you.
What was
Sir,
it?
immunodiagnostic test, what are the important things that we have to know about the
Okay,
immunodiagnostic test?
you have to know what are the immune diagnostic tests, like, and what are the antigen antibody reaction? What are the antigen antibody reaction, like agglutination, precipitation, and some are labeled like ELISA, or radioimmunoassay or chemiluminescence. Some are non-label tests like agglutination, precipitation, and the difference between the agglutination and precipitation, because in the last written there is a question that what is the difference between the agglutination and precipitation, and what is the principle of ELISA? These are the things actually you need to know. Apart from just go through the previous year question, written question is the main thing. Viva, you will very rarely asked in this topic. Just few of the written you have to solve. Any other
Okay,
question?
sir.
Do you have any other question?
Okay, sir. Thank you.
Welcome. Do you have any other questions? So what we have actually learned in this review class, that we will see immunology, or we will learn immunology as a whole, not to just memorize it. We have to correlate the things, and this is kind of math. There are formulas, and you can adapt, you can apply the formulas in everywhere. Like antibody, what are the antibody works? It can work as a ADCC. There is a placenta transfer, so that you can see that this is actually correlated, and it is not so tough. It is not so tough. If you study with tactics, it will be very easy. So this is the single message I want to share with you. So that's it. If you do not have any question, I will finish. Thanks for attending the class. It was interactive. And do you have any further question? If you do not, then I'm going to finish the class.